Hello, everyone, and welcome to today's live broadcast, European Pharmacopoeia and Complete TOC Oxidation for Purified Water and Water for Injection, presented by Tony Harrison. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by Beckman Coulter Life Sciences. Beckman Coulter Life Sciences develops, manufactures, and markets products that simplify, automate, and innovate complex biomedical testing. More than 275,000 Beckman Coulter systems operate in both diagnostics and life sciences laboratories on 7 continents. For over 75 years, our products have been making a difference in people's lives by improving the productivity of medical professionals and scientists, supplying critical information for improving patient health, and delivering trusted solutions for research and discovery. To learn more about Beckman Coulter Life Sciences, please visit www.beckman.com. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. Finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Tony Harrison, a Senior Marketing Manager at Beckman Coulter Life Sciences. Tony is experienced in water system total organic carbon, conductivity, ozone analysis, cleanroom environmental monitoring, and particle characterization. He has spent the last 15 years in applied metrology for the pharmaceutical and healthcare manufacturing industries. Tony held the convenorship of the ISO working group revising ISO 14698-1 and 2 for microbial control in cleanrooms. He served as the UK subject matter expert to the ISO working group that issued the 2015 revised versions of the ISO 14644-1 and 2 documents for cleanroom classification. These were at the heart of the aseptic manufacturing chapters of the European GMP and the USA cGMP documents. Tony was joint editor on the ISPE Guide to Ozone Sanitization of Pharmaceutical Water Systems and was also chief editor of the PHSS Best Practice Guide for Cleanroom Monitoring. Tony is a well-known international speaker and has provided educational seminars around the globe on TOC, liquid particle counting, ozone sanitization for water systems, and cleanroom monitoring. His complete bio is found on the LabRoots website. Please join me in welcoming Tony Harrison. I will now turn the presentation over to him.
Thank you very much indeed for that introduction. Hello, everybody. My name is Tony Harrison from Beckman Coulter Life Sciences. Today I'm going to discuss in this very short presentation the recommendations in the Chapter 2.2.44 of the European Pharmacopoeia that total organic carbon analyzers designed for measuring purified water and water for injection should fully oxidize the organic contaminants to carbon dioxide to release the organic carbon for accurate TOC measurement. Now, as I said, this is a relatively short presentation lasting approximately 20 minutes, depending on questions and answers. First, we'll discuss exactly what total organic carbon is, and then we'll move on to take a quick look at the recent changes to the European Pharmacopoeia chapter on water for injection and how this is increasing the importance of accurate TOC analysis. Then we'll examine one of the key problems stopping us carrying out root cause investigations on TOC excursions. And next, we'll discuss the European Pharmacopoeia chapter 2.2.44 on TOC and why it calls for complete oxidation of the organics for accurate TOC analysis. Finally, today, we'll take a look at the P8700 TOC and conductivity analyzer from Beckman Coulter in the light of the European Pharmacopoeia requirements for complete oxidation. So let's take a look at total organic carbon, just what it is and how it's actually measured. the FDA mandates 4 critical water quality attributes for water for injection and purified water. Inorganic contaminants should be measured, total organic carbon, endotoxins, and microbial colony-forming units should all be monitored. Total organic carbon and conductivity are the only real-time measurements amongst these 4 and can give almost instant warning of a potential contamination event. Now, total organic carbon measurement, it's a non-specific test. The results of a TOC analysis can tell you the amount of organic carbon in the water, but it cannot tell you exactly what the organic contaminants present are. TOC is in fact a measurement of the mass of carbon atoms present in organic material in the water, reporting TOC in units of mass per litre of water, i.e. micrograms per litre, or more frequently in the pharmaceutical industry, expressed as parts per billion. The carbon atoms are released from the organic molecule by breaking the molecule down through oxidation. The carbon forms into carbon dioxide, which in turn dissolves into the water, creating carbonic acid, which is very weak acid, but it elevates the water sample's conductivity. Before the TOC is measured, the TOC analyzer must first measure the amount of naturally occurring dissolved carbon dioxide in the water. And this is called the total inorganic carbon. And then the organics are oxidized to release the carbon they contain, and the total carbon is measured. By subtracting total inorganic carbon from total carbon, the level of total organic carbon in the water can be ascertained. Now, TOC analyzers that utilize conductivity to measure the organic carbon can also be used to measure conductivityone of the other parameters. However, the pharmacopoeias are very specific regarding calibration of conductivity analyzers, and the TOC analyzer itself would need to comply fully with their requirements. So, for example, the United States chapter on conductivity, USP 645, is very clear that the conductivity meter cell constant must be verified against a certified conductivity solution, and also that the meter electronics must be verified by the use of a certified calibration resistor. Since the changes to the European Pharmacopoeia chapter on water for injection in 2016, total organic carbon has taken on an even more critical role in detecting potential water contamination events. Although much work has been done in the last decade to harmonize the pharmacopoeias, the European Pharmacopoeia chapter on water for injection stood out because it specifically called for the use of a still for the manufacture of water for injection. Other pharmacopoeias merely listed the key quality parameters of water for injection, which are, of course, total organic carbon, conductivity, microbial colony-forming units, and endotoxin levels. However, with pressure to reduce carbon footprints and reduce manufacturing overheads, representatives from the United States Pharmacopoeia, the European Pharmacopoeia, and the Japanese Pharmacopoeia came together to survey the pharmaceutical industry regarding the quality of water manufactured by reverse osmosis, and in particular, the level of control these water treatment plants had over microbial colony-forming units. During the period from 2004 to 2010, the JP, USP, and EP gathered surveys on water for injection made from reverse osmosis from around the world. The results were surprisingly good, with many studies showing an excellent water quality matching water for injection standards, and with very good levels of microbial colony-forming units control. So in 2011, an expert workshop was formed with representation from the pharmaceutical manufacturing industry to consider changing the European chapter on water for injection to allow the use of reverse osmosis as long as the water produced, of course, met the key quality attributes of water for injection and showed high levels of microbial control. And finally, satisfied with their work, in 2016, the new chapter for water injection in the European Pharmacopoeia was adopted, allowing for the first time the use of reverse osmosis for the production of water for injection. Now, the working party who supported the European Directorate with their 2016 new chapter for water injection made very clear that they warned that reverse osmosis water treatment may well not be as robust as a still. And they highlighted the role of total organic carbon and conductivity analyzers as early detectors of potential impending microbial or even endotoxin excursion events. Naturally, any total organic carbon or conductivity excursion should lead to a root cause investigation. But many excursions are transient in nature, and it is often difficult to capture a sample of the contaminated water to carry out the root cause investigation. So here's an example. The problem is that TOC excursions, as I said, can often be transient events. Now, they're detected by the TOC analyzer online, but they disappear by the time the team comes to take the water samples for root cause investigation. As stated earlier, 1 parts per billion of carbon is equivalent to 1 microgram of carbon in 1 liter of water. So in the example here, typical example of a modern water for injection system, the TOC levels are commonly recorded at around 26 parts per billion. Now say, suddenly, a slug of total organic carbon is fed into the water system by accident, either from one of the points of use or from biofilm shedding, and this causes an excursion of 750 ppb on all the other points of use, equivalent to 750 micrograms of carbon in 1 liter of water. Now, the online TOC analyzer detects this and reports it as an excursion. And then the 750 ppb of micrograms of carbon now enters the water for injection storage tank, which in our example contains 1,000 liters of water for injection. So now the 750 micrograms of carbon is diluted in that 1,000 liters of water for injection and actually becomes 0.75 micrograms per liter of carbon, or 0.75 ppb. Then this diluted carbon is very easily destroyed by the UV lamp, which is commonly used for microbial control and water for injection systems as the water passes back out into the distribution loop. And now the TOC levels in the loop have returned to their safe low TOC value. But it's too late. During that excursion, levels of TOC at the points of use were outside the pharmacopoeia limits, and any batches being manufactured at that time cannot be released into the market. And as I said, what makes the situation worse is there's no way to carry out a root cause analysis to find out the likely sources of the TOC excursion, as all of the evidence has been diluted and destroyed. So there's no way to put a preventative action in place to stop recurrence of the excursion again at a later date, and this therefore could compromise future batches as well. So to the center of this presentation today, if online TOC analyzers are truly to be used to provide supporting batch release data, then there are some significant issues to consider, and not least of which is complete oxidation of the organic contaminants in the sample to ensure accurate TOC measurement. Now, this is an excerpt from the text of the European Pharmacopoeia chapter on total organic carbon, chapter 2.2.44. It emphasizes the importance of ensuring the complete oxidation of the organic molecules to ensure that all organic carbon present is released and measured accurately. Most TOC analyzers used to monitor pharmaceutical grade water use ultraviolet light to create hydroxyl radicals from the water, which in turn oxidize the organic material present, releasing the carbon atoms in the form of carbon dioxide. As I mentioned, the carbon dioxide dissolves into the water, creating carbonic acid, which elevates the conductivity, and this change in conductivity is measured by the TOC analyzer and used to calculate the amount of organic carbon present in the water. The problem is that as long, complex chain molecules are broken down during the oxidation process, they may form organic acids as intermediate species, which in fact will contribute more strongly to the water conductivity than the final carbon dioxide does. If the TOC analyzer does not complete the oxidation of the organic molecule, then one of two things may happen. Either the carbon remains bound inside the organic and cannot be measured, so that TOC is underreported, or the TOC analyzer takes a conductivity measurement before the complete oxidation when the organic acids are present, and it overreports the amount of TOC in the water. Either way, assuring complete oxidation to carbon dioxide is the only way to measure TOC accurately. Now, some TOC analyzers have a fixed oxidation time, and the water continuously flows through them and is only exposed to the ultraviolet light for short periods of time. Now, these can struggle to report TOC levels accurately. The International Conference on Harmonization is an expert working group with representation from the European, United States, and the Japanese pharmacopoeias. And in their ICH-Q2 document, Validation of Analytical Procedures, they recommend a system suitability test to determine the robustness of an analyzer's measurement. Should a TOC analyzer with fixed oxidation times be calibrated with a relatively easy to oxidize TOC standard such as sucrose, then it will likely not report accurately if challenged with a relatively difficult to oxidize TOC standard such as the SDBS system suitability test as recommended in the Japanese Pharmacopoeia chapter on TOC. The fixed flow path and fixed exposure time to the UV light will mean that the SDBS will not be fully oxidized and much of the carbon will remain unmeasured. Now, some TOC analyzers employ persulfate as well as the UV light. Now, the persulfate extends their measurement range, and in the presence of UV light, the persulfate becomes a very, very strong oxidizing agent, enabling the TOC analyzer to oxidize relatively large amounts of TOC in a short time. However, in designs employing gas-selective membranes as part of the measurement cell, this can present some challenges. If too much persulfate is added, then there is a danger that bubbles may form in a sample, and these bubbles can occlude or block the measurement membranes, leading to underreporting of TOC levels. And equally, if too little persulfate is added, then the TOC may not be fully oxidized, and once more, the TOC analyzer will underreport the TOC content in the sample. In this paradoxical situation, the analyzer user somehow has to know in advance the value of TOC in the sample so that they can configure the persulfate dosing correctly on the analyzer before they carry out the measurement. So, let's take a look at the PW700 TOC and conductivity analyzer from Beckman Coulter and see how it can help compliance to the European Pharmacopoeia requirements. Now, this analyzer is not a general purpose analyzer. It is designed specifically for TOC analysis on purified water and water for injection. And the PAT TOC analyzer from Beckman Coulter employs a variable oxidation time combined with a dynamic endpoint detection to determine when the sample oxidation is complete. Firstly, an aliquot of the water being sampled is tracked in the measurement cell. There's a single conductivity sensor that measures the conductivity of the sample, which is representative of the TIC, total inorganic carbon in the sample. A UV lamp is then turned on to oxidize the organics in the sample. As the molecules are oxidized, the released organic carbon in the sample turns to carbon dioxide, which dissolves and increases the conductivity of the trapped aliquot of water. The PW700 monitors the change in conductivity of the sample, and when the conductivity stops changing, the PW700 knows that the analysis endpoint has been reached and the sample has been completely oxidized. Now the sample contains both the TIC, total inorganic carbon, and the total organic carbon, i.e. it's got total carbon. Total carbon conductivity is measured using the same conductivity sensor used to measure the inorganic carbon, and one is subtracted from the other to calculate total organic carbon. By using a single sensor to measure both the inorganic and total carbon, the effects of any slight conductivity sensor drift over time on the accuracy of the TOC calculation is eliminated, as the conductivity sensor drift would apply equally to both the inorganic carbon and the total carbon measurement, and thus cancel out. As the oxidation time of the P8700 is variable and controlled by dynamic endpoint detection, the analysis of harder to oxidize materials such as SDBS as recommended in the system suitability test in the Japanese Pharmacopoeia does not cause a problem. The P8700 simply extends its analysis time, continuing to oxidize until it detects the sample conductivity has stopped changing and the oxidation is complete. Now, of course, these analyzers use ultraviolet light to oxidize the organics, so the level of the ultraviolet light being emitted from the UV lamp is critical. And in the P8700, it is measured to ensure that it is sufficiently powerful to oxidize the sample, and in the event of a lamp failure, a second standby UV lamp is turned on to ensure complete oxidation. Now, as I mentioned earlier, TOC or conductivity contamination events can be transient in nature. As I mentioned, a system that's experiencing gradual biofilm buildup may experience TOC excursions, particularly just after the sanitization cycles as biofilm sloughs off the inside of the pipework. This excursion can quickly disappear either by being diluted as it passes into the loop storage tank, or broken down by UV lamps connected to the pipework distribution network put in place to discourage microbial buildup. However, this rapid disappearance may mask the increasing biofilm buildup until there is a serious breakdown of system control and runaway microbial contamination. To help with root cause investigation, the P8700 can be programmed to capture a water sample should a TOC excursion be detected, so that the sample can be further analyzed to get to root cause, hence supporting kappa. Now, one of the additional advantages of dynamic endpoint detection for complete oxidation in the P8700 from Beckman Coulter is that it can provide an indication of the oxidation curve profile during each TOC analysis. Each organic will have a slightly different oxidation profile, so a change in the type of profile combined with a change in TOC levels may provide insight into a change in the water system organic chemistry, suggesting a potential degradation of the reverse osmosis system integrity, prompting the user to investigate further and consider corrective action to prevent a large-scale contamination event and to put in place preventative measures to avoid the recurrence of the problem in the future. So we've learned today that TOC and conductivity analyzers can be strong allies to help us keep our water systems under control. We've learned how calibration is important and how complete oxidation is recommended by the European Pharmacopoeia for accurate TOC analysis. So let's just summarize how the P8700 TOC and conductivity analyzer can support you through the challenges of moving to water for injection release. So as leading edge indicators of potential changes in water system health, online TOC and conductivity analyzers are taking an ever-increasing importance in preventing contaminated purified water and water for injection from mixing with expensive drug product during manufacturing. The ability to report changes in water organic chemistry combined with TOC transient excursion capture provides more support for root cause investigations before the purified water or the water for injection exceed the pharmacopoeial limits. Complete TOC oxidation to release all of the organic carbon as carbon dioxide, even with long-chain, difficult to oxidize organic molecules, as per the European Chapter 2.2.44, is paramount to accurate TOC measurements if they're to be made. So now that concludes my talk today. I'd be happy to take any questions, but I'd mention that there should be a poll question posted on your screen to ask you if you'd like to receive my white paper on this subject. If you would like to receive that paper, please click on the yes and submit it. So now I'm happy to take questions.
Thank you, Tony, for that informative presentation. It is time for Q&A, so if you have a question you'd like to ask Tony Harrison, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for, so let's get started. Our first question is, can TOC be diluted?
Yeah, actually, that's a very interesting question and very pertinent, really. It can certainly be diluted. And although we talk about parts per billion of total organic carbon, in fact, it is a measure of mass of carbon atoms in micrograms per liter. So if a TOC excursion happens in the water loop, when it enters into the loop storage tank, it gets diluted by the large volume of water in the storage tank and is effectively removed, and subsequent water samples taken to support root cause investigations will not contain sufficient concentrations of the organic contamination to be effective. And that's why the P8700 automatically traps a sample of the contaminated water for further analysis, thus supporting root cause investigation.
Why does the European Pharmacopoeia call for complete oxidation of the organic contamination?
So, as I mentioned in the presentation there, the total organic carbon is measured by oxidizing the organic material to carbon dioxide and then measuring the increase in conductivity that this actually creates. So if you don't completely oxidize all of the organic contamination to carbon dioxide, then you won't accurately measure the TOC in the water. So flow-through TOC analyzers, which have a fixed oxidation time, can struggle to measure TOC excursions. That's why the P8700 traps the sample for analysis and monitors the change in the sample conductivity until it stops changing, i.e. all of the organic has been completely oxidized to give an accurate measurement.
What happens to the effectiveness of TOC analysis if the UV lamp fails prematurely?
Yeah, that's another good question. So the UV lamp is in fact the TOC analyzer engine, if you like, and without the UV lamp there's no oxidation of the organics and no TOC measurement. Now, unbelievably, most TOC analyzers do not monitor the UV lamp light output. So if the lamp fails prematurely, then the analyzer will not measure TOC and won't report any TOC excursions. Now, this risks the problem of drug product being released onto the market out of specification, leading to subsequent product recall when the error is detected by the QC lab analysis. And that's why the P8700 monitors the UV lamp light output and has a standby UV lamp which automatically switches in if the main lamp fails.
I would like to once again thank Tony Harrison for his presentation. Do you have any final comments?
No, just to thank everybody for listening today, and I hope it was informative.
Well, thank you once again. I would also like to thank our sponsor, Beckman Coulter Life Sciences, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.
Thank you.